We take the physical stability of our bodies totally for granted.
Oh, absolutely.
I mean, you wake up, you look in the mirror, and you just instinctively assume, like, your skin is still skin, your liver is still a liver.
Like, the adult gut is still an adult gut.
Exactly. But what actually stops your adult cells from suddenly, forgetting their specific identity and sliding backward in time, like reverting right back into an embryonic state?
It is, honestly, one of the most profound biological mysteries we face, because we intuitively think of adult cell identity as this locked door. Once a cell matures, the lock turns and it's permanent.
But it's not permanent, is it?
No, not at all. The reality is that the door requires constant, active, massive metabolic effort to keep shut. If the cellular machinery holding that door closed slips even a fraction of an inch, the cell doesn't just die. It can literally time travel back to its fetal days.
Wow. And that biological time travel is our exact mission for this deep dive. Today, we are unpacking a phenomenal paper published in Cell Stem Cell from Shinya Yamanaka and his colleagues.
Yeah. And Yamanaka, of course, famously won a Nobel Prize for figuring out how to force adult cells back into an embryonic state.
Right. The induced pluripotent stem cells.
Exactly. But today's source material actually flips the script on that entirely.
Yeah, we are looking at how a single, rather obscure piece of cellular machinery actively prevents our gut cells from making that exact time-traveling leap naturally, like every single second of the day.
And the underlying biology here is incredibly elegant. The researchers focused on a non-canonical translation initiation factor called EIF4G2.
Okay, wait, EIF4G2?
Yeah. And their work maps out in staggering detail how this one specific protein safeguards the identity of adult intestinal stem cells.
Okay, let's establish the baseline mechanics here before we look at what happens when things go wrong. We know that cells maintain their identity through translation, right?
Right, taking the mRNA transcripts and turning them into functional proteins.
But you specifically use the term non-canonical translation initiation factor for EIF4G2. How does that differ from the standard everyday translation happening in the cell?
So the vast majority of proteins in your cells are built using canonical translation, which relies on a piece of machinery called EIF4G1.
Okay, so G1 is the standard one.
Exactly. That standard machinery binds to the cap of the mRNA, stands along the transcript until it finds the start codon, and begins building the protein. But EIF4G1 has a major limitation.
Which is?
Well, if the mRNA transcript has highly complex secondary structures, like tight RNA hairpins or incredibly dense regulatory sequences before the actual start code, the standard EIF4G1 machinery physically stalls out.
Oh, it just gets stuck.
Yeah, it just can't get through.
So if I'm picturing this, the standard machinery is great for reading a straightforward linear genetic code, but it just jams up if the code is tangled or heavily encrypted.
That is exactly where EIF4G2 comes in. It's an alternative, highly specialized translation factor designed specifically to bypass those complex structures.
Okay, so it reads the encrypted stuff.
Right. It targets mRNAs that have long 5' untranslated regions or 5' UTRs, and also upstream open reading frames.
Which are what, exactly?
They are essentially dummy start codes that confuse the standard machinery. But EIF4G2 has the biochemical capability to navigate those dense, tangled 5' UTRs and ensure those specific proteins actually get built.
Okay, I want to push back on something here, though. If EIF4G2 is a translation factor-like, a core piece of the machinery required to build proteins, shouldn't knocking it out of the cell just trigger a total catastrophic failure?
That's a very logical assumption.
I mean, if you stop translation, the cell should just shut down and die immediately, right?
It's exactly what the researchers tested first. To see if the cell would just die from a lack of protein, they used the sunset assay.
Sunset assay. What does that stand for?
It stands for surface sensing of translation. It's a really brilliant technique. They pulsed the cells with puromycin.
Wait, puromycin is an antibiotic, right? How does that measure protein synthesis?
Well, puromycin physically mimics a piece of transfer RNA. So as the cell's ribosomes are actively building new proteins, they accidentally grab the puromycin and incorporate it directly into the growing protein chain.
Oh, that's clever.
Yeah. The researchers can then use a highly specific antibody to detect that puromycin. It gives you a perfect real-time snapshot of the total global protein synthesis happening in the cell.
Okay, so using a factory analogy, it's like throwing a highly visible fluorescent dye into a factory's raw materials just to see how much total product is coming off the main assembly line.
Exactly. And the sunset assay showed that global protein synthesis was totally fine.
Really? Totally fine.
Yeah. When they knocked out EIF4G2, the cells weren't dying from a lack of overall protein. The main EIF4G1 factory was still churning out product.
Wait a minute. So if global translation is continuing without a hitch and the cells aren't just collapsing, what is this specialized EI4G2 machinery actually doing that is so critical?
To figure that out, they couldn't just look at global protein. They had to look at every individual mRNA. So they employed ribosec.
Which is ribosome profiling, right?
Yes. This technique basically freezes the ribosomes in place and sequences only the tiny fragments of mRNA that are physically protected inside the ribosome.
Oh, so it tells you exactly what is being built in that exact moment.
Right. It lets you calculate the exact translation efficiency of every single gene. And the ribosec data revealed that while most mRNAs were completely unaffected, a very specific, heavily guarded subset of mRNAs saw their translation plummet to near zero.
Okay, I just want to pause and make sure we have this foundational logic locked in before we move to the physical tissue. Let's do a quick mini recap.
Sounds good.
So, bullet 1. EIF4G2 is a specialized cellular printing press, not the main one.
Correct.
Bullet 2. It specifically translates mRNAs with complex 5' UTRs. And bullet 3. Losing it doesn't stop overall protein production, just a targeted subset.
Spot on, and that sets the stage for a highly targeted crisis rather than a global cellular collapse, because the proteins that aren't getting printed happen to be the master regulators of the cell's identity.
Which brings us perfectly to the next huge question. What happens to a living, breathing tissue when it loses those specific, heavily guarded proteins?
Well, the researchers looked at the intestine, which makes perfect sense, because the lining of the gut is one of the fastest renewing tissues in the human body.
Right. The turnover rate is incredible.
It is. The gut lining is structured with these deep valleys called crypts. And at the very base of these crypts are the LGR5 positive intestinal stem cells.
The LGR5 cells.
Yeah, these adult stem cells are the engine of the gut. They constantly divide and push new cells upward to form the villi, which are the structures that actually absorb your nutrients.
Okay, so to test what happens when EIF4G2 goes missing in these vital stem cells, the researchers used a genetic tool called the CREER system, right?
Yes. The CREER system is one of the most elegant tools in molecular biology.
Break that down for us.
So CREE is an enzyme that cuts specific sequences of DNA. But in this system, it's physically fused to an estrogen receptor, the ER part. Because of that bulky receptor, the enzyme is trapped in the cytoplasm of the cell. It can't get into the nucleus to touch the DNA. It just floats there harmlessly.
Until you introduce the trigger.
Right. You inject the mouse with tamoxifen. The tamoxifen binds to the receptor, causes a shape change, and suddenly the CREE enzyme rushes into the nucleus and snips out the target gene.
Which in this case is EIF4G2.
Exactly. It's essentially a remote-controlled genetic deletion. They ran this using a systemic promoter, ROSA26, which deletes the gene everywhere, and a gut-specific promoter, VIL1, which ensures the gene is only deleted in the intestinal tissue.
Okay, and here's where it gets really interesting. The phenotype, like the physical result of this remote-controlled deletion, is staggering.
It really is.
Once EIF4G2 is gone, the adult LGR5 stem cells completely vanish from the crypts.
Yep. And secretory cells, like the Panath cells that provide antimicrobial defense and the goblet cells that produce mucosal lining, they just drop off a cliff.
But the intestine itself doesn't die.
No, it survives by undergoing a massive systemic identity shift. The tissue adopts what the researchers termed a durable fetal-like regenerative state.
A fetal state.
Yeah. The adult LGR5 marker is entirely gone, and suddenly a marker called BMI1, which is characteristic of fetal development, surges across the entire crypt base.
Man, to really understand how deep this time-traveling shift went, the researchers utilized single-nucleus multiome profiling, or sen-multiome, right?
They did. And getting transcriptomic RNA data and ATAC-seq chromatin accessibility data from a bulk tissue is one thing, but doing it from a single nucleus simultaneously is notoriously difficult.
Because the chemical prep usually ruins the RNA, right?
Exactly. Opening the chromatin for ATAC-seq totally degrades the fragile RNA. It is incredibly technically demanding.
But the payoff is massive.
Oh, absolutely. Because they captured both readouts from the exact same single nucleus, there is zero guesswork. They could perfectly correlate which genes were physically open and accessible with which RNA transcripts are actually being produced.
And what did that one-to-one correlation reveal about these newly fetal-like cells?
The sen-multiome data showed a dominant YAP-TED-driven signature.
Yeah.
Right. YAP is a highly conserved protein pathway that gets massively upregulated during fetal tissue development and also during extreme tissue regeneration.
Okay, but hold on. Let me play devil's advocate here for a second. Couldn't this just be the cells getting intensely inflamed?
How do you mean?
Well, losing your primary adult stem cells and all your secretory cells has to be a traumatic event for the tissue, right? Whenever tissue is injured, say by radiation or a toxin, it temporarily reverts to a fetal state, declares a state of emergency, survives the stress, and then eventually goes back to normal.
Right, that's the standard injury response.
So how do we know this isn't just a standard temporary inflammatory response?
The researchers anticipated that exact critique. They ran their transcriptomic data against the MSigDB hallmark gene sets.
Which is what, a database?
Yeah, it's a comprehensive database of known cellular stress and inflammatory pathways. They specifically looked for canonical inflammatory drivers, like interferon alpha signaling or the integrated stress response, the ISR.
And they weren't there.
No. Well, they found a minor stress signal in the secretory cells, which makes sense as they were failing to mature and dying off. But the ISR and canonical inflammatory pathways were not the driving force behind the massive shift in the stem and absorptive cells.
Wow.
Furthermore, in the Vilwen-Krier mice, where the knockout was restricted just to the gut, the mice maintained their body weight and survived for months.
So it wasn't just a temporary panic response to an acute injury?
Exactly. This was a stable, chronic, permanent reprogramming. The tissue established a totally new equilibrium.
Okay, let's unpack this with another mini recap. So bullet 1, removing EIF4G2 deletes adult LGR5 stem cells. Bullet 2, the gut compensates by permanently reverting to a BM1 positive fetal-like state.
Yep.
And bullet 3, this is a stable identity shift, not just a passing inflammatory stress response.
That's right. Which leaves us with the critical mechanical gap in the paper.
Right. How does missing a protein translation factor out in the cytoplasm cause the physical DNA architecture inside the nucleus to rewrite its own identity?
This is the epigenetic bridge. This is where the paper pulls everything together.
I love this part.
So to find that bridge, the researchers went back to the RiboSec data. They looked at that specific list of complex mRNAs that EIF4G2 was uniquely responsible for translating.
And what did they find?
The dominant targets that plummeted when EIF4G2 was lost were transcripts encoding crucial chromatin regulators, specifically two proteins called CREBP and EP300.
Which are collectively known as the KAT3 coactivators, right?
Yes.
Okay, let's unpack what the KAT3 coactivators actually do. Think of them less as simple on-off switches and more like heavy-duty epigenetic structural engineers.
That's a great way to picture it.
Yeah, so your DNA is tightly wrapped around protein spools called histones, tightly packed together in the dark. If a gene is tightly spooled, the cellular machinery can't physically reach it to transcribe it.
Right.
So the KAT3 engineers walk along the DNA and attach bright chemical tags, specifically acetyl groups, like H3K27C ACI onto those spools.
And the physics of that acetylation are crucial here. DNA naturally has a negative charge.
Okay.
When KT3 attaches an acetyl group to the histone, it alters the local electrical charge, causing the histones to physically repel each other.
Oh, so they push apart.
Exactly. It wedges the tightly packed chromatin wide open, creating a physical space for the transcription machinery to enter and read the DNA. Those H3K27 ACK marks literally define the active enhancer regions of a cell's identity.
Okay, so let's trace the exact causality. Without EAF4G2, the complex mRNAs for the KAT3 engineers never get translated.
Right.
The KAT3 protein levels collapse. Without KAT3, the H3K27 acetyl marks are no longer being actively placed on the DNA.
The chromatin just snaps shut.
Just snap shut.
But, and this is key, it snaps shut in a highly locus selective way.
Yeah.
The SMIN multiome data showed that the genes maintaining adult intestinal identity genes driven by the Wnt and Notch pathways, like ASCL2 and Notch1, completely lost their H3K27 ACK marks.
So the adult genes close.
The chromatin condensed and the adult factory programs plunged into darkness. But simultaneously, the chromatin surrounding the fetal and regenerative genes, like TAXTD2, somehow gained accessibility.
Wait, they opened up?
Yes, they opened up, and the fetal YAPI-TED program roared to life.
That is wild. And to physically prove that those active enhancer marks were moving from adult genes to fetal genes, the researchers utilized CT and tag sequencing.
Yes. CUT and tag is an incredible evolution of older chromatin mapping techniques.
It stands for cleavage, under targets, and tagmentation, right?
Exactly. Instead of grinding up the whole genome and hoping you pull down the right fragments, you send in a targeted strike team.
I like that visual.
You introduce an antibody that binds specifically to that H3K27 axial mark. Then you introduce a tethered TN5 transposase enzyme.
The TN5 transposase.
Right. The transposase binds to the antibody, and it literally cuts the DNA right there, physically inserting the sequencing adapters directly into the open chromatin adjacent to the mark.
So it maps the exact coordinates of every open active gene across billions of base pairs?
And it confirmed that the adult enhancers were silenced and the fetal enhancers were activated.
But this is biology, so correlation isn't enough, right?
Yeah.
We have to prove that the loss of the KAT3 engineers is the direct, exclusive cause of this massive fetal reversion.
Absolutely. And that is where they utilized a brilliant pharmacological intervention. They took completely normal, healthy adult intestinal organoids, which are essentially 3D mini-guts growing in a dish.
Okay.
And they treated them with a small molecule drug called A485.
A485.
Yeah, A485 is a highly specific catalytic inhibitor of the KAT3 proteins, CREBP and EP300. It doesn't delete the gene, it just chemically paralyzes the engineers so they can't place the acetyl marks.
And the result of chemically paralyzing KAT3 was a flawless mimic of the genetic knockout, wasn't it?
Spot on. The healthy adult organoids immediately lost their mature markers, their complex crypt structures disintegrated, and they physically collapsed into these smooth, cystic, fetal-like spheroids.
It perfectly closed the causal loop. So just to do our third mini recap here.
Let's do it.
Bullet 1. EIF4G2's main job is translating KAT3 proteins. Bullet 2. KAT3 proteins place H3K27 AC marks on adult genes to keep them active. And bullet 3. Without KAT3, adult genes turn off and fetal YPTED genes turn on.
Perfectly summarized.
Okay, let's tie this together with one final massive question. If this EIF4G2 KAT3 pipeline is so fundamentally essential for maintaining cellular structure, does every single cell in the gut require it just to survive?
That's the logical next question.
I mean, what happens if you take this machinery away from a cell that is already in a fetal state?
Well, the researchers grew genuine fetal intestinal spheroids derived directly from E14.5 mouse embryos. And they triggered the exact same Crier genetic knockout of EIF4G2 in these truly fetal cells.
Did the fetal cells just implode?
Well, they ran the biochemical assays. Just like in the adult cells, knocking out EIF4G2 caused the KAT3 protein levels to plummet.
Okay.
And just like in the adult cells, the H3K27 AC epigenetic marks dropped drastically. The exact same biochemical failure occurred.
But physically?
Phenotypically, the fetal spheroids didn't care at all.
Wait, really?
They survived perfectly. They continue to propagate and grow robustly, entirely unfazed by the loss of this machinery.
They just kept going. Wow. It really highlights the sheer metabolic cost of being an adult.
It does.
If an adult cell is like balancing a heavy broom vertically on the palm of your hand, it requires constant, active, energy-intensive micro-adjustments just to stay upright.
That is a perfect analogy.
And that constant adjustment is what the authors call translational buffering. The EIF4G2 printer continuously churning out KAT3 engineers to physically force the chromatin to stay in an adult conformation.
And when that translational buffering fails, gravity simply takes over. The broom falls, and it lies completely flat and stable on the floor.
And that floor is the fetal regenerative state.
Exactly. Fetal identity is the robust, stable default baseline of biology. It naturally lacks those complex adult markers, so it simply doesn't require the massive epigenetic overhead of KAT3 to maintain itself.
So our fourth mini recap. Fetal cells naturally lack adult markers. They don't rely on KAT3, and when you delete EIF4G2 in them, they survive perfectly, meaning the adult tissue requires active, constant epigenetic maintenance that fetal tissue doesn't need.
Right. The adult state is entirely synthetic, in a way. It is an active rebellion against the biological default.
Which fundamentally reframes how we have to look at human disease.
Absolutely.
Exactly. And this is where we bring it back to you, the listener. So what does this all mean for you? Why should you care about understanding this obscure, non-canonical translation factor? What does this mean for the future of medicine?
Because this specific mechanism, the collapse of high-energy adult identity into a durable, low-energy fetal state, is driving some of the most intractable challenges in human health.
Like what?
Well, look at chronic inflammatory diseases like inflammatory bowel disease, or severe tissue damage from aging, or even the very origins of cancer. When adult tissues are subjected to repeated, relentless damage, their translational buffering eventually fails. They drop the broom, they revert to these durable, fetal-like regenerative states just to survive the harsh environment.
They stay alive, but they completely lose their mature functional capacity.
Exactly. They survive the endless state of emergency, but they permanently forget how to do their actual day job in the organ.
And Yamanaka's team has just handed us a high-resolution blueprint of the exact bridge that breaks during that process.
They really did. They mapped the direct causal chain from a specific protein translation factor straight to the epigenetic coactivators, right down to the physical restructuring of the DNA.
Which is huge for potential treatments.
Definitely. If we understand precisely how this bridge collapses, the next step is figuring out how to pharmacologically reinforce it. We could potentially design therapies that help chronically damaged tissues maintain their adult buffering.
Preventing a stressed intestine from tipping over the edge into a fetal-like state, which, is often the precursor to cancer.
Exactly. The implications are massive.
It is truly mind-blowing to realize that your body isn't just statically existing. It is actively fighting to maintain its identity every single second against the biological pull of its own developmental past.
The complexity of how our cells actively remember who they are is endlessly fascinating.
It really is. Thank you for joining us on this deep dive into the source material. You are now armed with the mechanics of one of the most cutting-edge biological insights in the stem cell field today.
That's been a great discussion.
And I want to leave you with a lingering thought to ponder as you go about your day. If the trillions of adult tissues currently making up your body are actively suppressing their fetal states every single second by continuously printing these highly specific epigenetic structural engineers, what other evolutionary or developmental ghosts are hiding in the architecture of your DNA right now, just one missing protein away from waking up?